Welcome friends and welcome to my channel Sequential Biology, and we have talked about virus. In the first classes we have discussed about property and the discovery of viruses, and in the second classes of this viral series we have talked about the structural component of viruses, their envelope, their capsid and their nucleic acid. And today we will discuss about Baltimore system of classification of virus. Okay, so before going to classification we should know few important terminology. Okay, and here we can see a DNA, and the DNA have two strand, and one is 5' to 3' strand. The one strand orientation is 5' to 3' and another is 3' to 5' strand. Okay, DNA is double stranded, showing the 5' region there are present phosphate group, and in the 3' region there will be present OH group of one strand, and its another strand is 3' OH and 5' phosphate. That is, the two strands are usually anti-parallel. This 3' to 5' strand is served as template. That is, during a time of transcription this strand will be transcribed. Okay, so mRNA will be synthesized from this strand. So this mRNA have complemented a base sequence with this template strand. Okay, and this 5' to 3' strand of DNA have the same base sequences with this mRNA, except thymine. The thymine is not present in case of mRNA. Okay, so mRNA has the complementary base sequence with the template strand of DNA. The template strand of DNA is 3' to 5' strand, and this is the template strand, and since this strand is transcribed, so the mRNA have complementary base sequences with this template strand. Okay, in virology the mRNA is said to be plus configuration. So this mRNA is said to be plus configuration, that is can directly translate to form the protein. Okay, so the mRNA undergoes the translation, and you know the mRNA contains many genetic code, and on the basis of this code the amino acid will be arranged during process of translation, and this will synthesize this protein. So this mRNA which directly translate to form the protein. Okay, so this mRNA is called as plus strand. The complementary strand of plus ssRNA, is called as minus ssRNA, and the minus ssRNA is actually called as antisense strand. Sometimes in case of viruses the few RNA can be found, and this RNA is complementary with this original RNA, and this is regarded as the minus strand. That is, the mRNA which directly produce the protein are called as plus strand. The mRNA which directly involved in protein synthesis are called as plus strand. Okay, and this mRNA is called as this RNA is called as antisense RNA, and this RNA is regarded as sense RNA, right? And this antisense RNA is called as minus strand in virology. Okay, so in virology mRNA is said to be plus configuration. They can directly translate to form the protein, and the complement of this plus ssRNA is called as the minus strand. Then the plus ssRNA undergoes the translation to form the protein, but from this plus ssRNA the transcription by process of transcription the minus ssRNA can be formed in few viruses. Then this is complementary to plus ssRNA, and this strand is regarded as minus strand. Now Baltimore have classified the viruses into 6 different classes. The class 1 viruses are double stranded DNA viruses, and you know that in viruses the single stranded DNA can also be found, and the double stranded RNA can also be found. Okay, so the class 1 viruses are double stranded DNA containing viruses. Example of these class 1 viruses are herpes virus and the pox virus. And class 2 viruses, the single stranded DNA viruses, then the double stranded DNA viruses, and next the single stranded DNA virus. Okay, the 5X174 bacteriophage and the chicken anemia viruses are grouped under class 2 viruses. Class 7 viruses are double stranded DNA viruses. Okay, first we have written the DNA and next RNA. Okay, so these double stranded DNA viruses replicate via an RNA intermediate. The double stranded DNA virus but replicate via an RNA intermediate. This is an unusual replication. So class 7 viruses, when they replicate via an RNA intermediate. This is very important. and later we will discuss about the replication process of these different classes. Okay, so these double-stranded DNA viruses, but they replicate via an RNA intermediate. This is an unusual replication. Actually, these are the partially double-stranded DNA. Example of this class 7 viruses are hepatitis B virus. Okay, next, class 3 viruses are double-stranded RNA viruses, and this is an unusual type of RNA, double-stranded RNA, and example of this double-stranded RNA viruses are Rio virus. Class 4 viruses, the single-stranded RNA containing viruses. The single-stranded RNA, this is plus RNA plus ssRNA, and this plus ssRNA directly involved in protein synthesis. Example of these class 4 viruses are polio virus, and whose RNA directly involved in protein synthesis. Next, class 5 viruses are single-stranded RNA viruses, but their strand is negative. Okay, so this strand is not directly involved in protein synthesis. This is complementary strand of ssRNA, or this is complementary of the plus ssRNA, and mRNA synthesized via an RNA dependent RNA polymerase. So what is this? The RNA which is not directly involved in protein synthesis, then this is called as minus RNA. Okay, but minus or minus ssRNA. So minus ssRNA. So it is very important for production of plus ssRNA. So this plus ssRNA will be transcribed. This plus ssRNA will be transcribed from this minus ssRNA, and this required an enzyme which is known as RNA dependent RNA polymerase. So RNA dependent RNA polymerase. RNA dependent RNA polymerase causes transcription of this minus ssRNA. RNA dependent RNA polymerase. Okay, usually RNA polymerase causes transcription of DNA, but RNA dependent RNA polymerase also causes transcription of RNA from another RNA. The production of RNA from any genetic material is known as transcription. This class 5 viruses possesses minus ssRNA as their genetic material, and this is complementary strand of the ssRNA or mRNA are synthesized by RNA dependent RNA polymerase. Example of this class 5 viruses which contains minus ssRNA as their genetic material are influenza and rabies viruses. And the last class of this Baltimore system of classification is the class 6 viruses. They are also called as retroviruses, and these retroviruses have positive strand ssRNA but replicate via double-stranded intermediate by enzyme RNA dependent RNA polymerase, and they also use reverse transcriptase. Certain cancer causing virus and the HIV, they are included under these class 6 viruses. Okay, so David Baltimore have tried to classify the viruses by following way on the basis of their genetic material, and these are very important to remember, that which viral genetic material is plus ssRNA containing genetic material and which is negative ssRNA containing genetic material. These are very important. And what are the example of these retroviruses? This is also important. Next video we will discuss about reproduction of virus, their replication. It is how they reproduce inside a host cell. So thank you, and that's it.